In this video, I'm going to talk about the special type of cell division known as meiosis. We've already talked about mitosis, which is the type of cell division that creates two identical daughter cells from one parent cell. And mitosis is used for making more cells for growth or for replacing and repairing damaged cells in multicellular organisms. Mitosis produces diploid or two-end cells, and these are often called somatic cells or body cells. They contain all the DNA of the original parent cell. In comparison, meiosis is used to create gametes, which are also known as sex cells, things like sperm and egg that are used for sexual reproduction. Gametes are produced by special cells. Not every cell can do meiosis, but germ cells found in the gonads, those cells can do meiosis. And the daughter cells created by meiosis contain only half the DNA as the parent cell. So they are known as haploid cells, or a chromosome number of N. Before we get into the details on meiosis, we need to talk about homologous chromosomes. Your diploid cells all have two copies of each chromosome because one copy is being received from each parent in the creation of that new organism, that offspring. This pair of matching chromosomes are known as homologous chromosomes, and you see the homo prefix, which meaning the same, as part of that word, because each chromosome in that pair have genes for the same traits, but the genes themselves are not always the same. For example, the same trait would be something like hair color in humans, but one chromosome in a homologous pair may have the gene for blonde hair, while the other one has the gene for brown hair. So the genes may not be the same, but the trait is between homologous chromosomes. And because of these differences in genes, we get more genetic diversity present. Just giving you an example from the human chromosome, this is a human karyotype, all the chromosomes found in a somatic, a body cell of a human. And so they're put together in pairs, and in each pair, one of those chromosomes would have come originally from the mother, and the other one would have come from the father. But the chromosomes themselves can be paired up. We can see they match in size, and then if you are doing more detailed work, you'll see they have the same genes on there for the same traits that those genes are covering. Because the cells that result from meiosis have half the number of chromosomes, it's a type of reductive cell division. It's reducing the number of chromosomes. You'll find that the stages of meiosis are very similar to those of mitosis, which you've already studied, but they do happen 2 times because we have to have this reduction in chromosomes. Overall, meiosis 1 ends up with a separation of these homologous chromosomes, and meiosis 2, the second time through the process, ends up with a separation of sister chromatids. So meiosis is going to start with prophase 1. DNA, of course, has already been replicated in interphase, and the DNA will condense into chromosomes just like you see with mitosis, with 2 sister chromatids held together by the centromere. In prophase 1 of meiosis, these homologous chromosomes will pair up and form what is known as a tetrad, a group of 4 chromosomes, through a process called synapsis. So on this illustration, here's our tetrad, our 2 chromosomes or 4 individual strands. These homologous chromosomes may actually trade pieces of DNA in a process called crossing over when they are in this tetrad position. They are very close together at this point. And this crossing over, exchanging of genes, actually leads to greater genetic diversity. So the second illustration is showing you that we've had a little bit of trading going on here. And so this chromosome, which may originally have come from the father, for example, now contains a few genes on this strand at least that originally arrived with the mother. In metaphase 1s, your tetrads line up along the equator of the cell, so they stay together as pairs. And they are randomly arranged, meaning it doesn't matter whether it comes from one parent or the other on whether they're to the north or to the south on the equator. So if we take the red as being chromosomes originally from the mother parent, we can see that 2 of the homologous chromosomes on this pair, on the north side are going to be from the mother, but 1 is going to be from the father. And of course, they're going to move to opposite poles at the next stage, and so that again mixes things up even more. Our illustration here is showing us we have 6 chromosomes arranged in 3 tetrads. Remember that, we're starting with 6 chromosomes. In anaphase 1, these homologous chromosomes are separated. That's what's being pulled to the opposite ends. But the sister chromatids remain attached, so we still have duplicated chromosomes. In telophase 1, the cell divides to create 2 haploid cells, so there's only 3 chromosomes in each cell, even though they're duplicated, because now we have reduced the chromosomes, but we still have these duplicates being held together by chromatids that have to be taken care of in the next stage. There's no interphase after telophase. The cell will go right into prophase 2 and do the second half of meiosis. Because there's no interphase, DNA is not replicated, and our chromosomes are going to remain tightly coiled up or condensed. We do get some new spindle fibers forming, though, in order to move these chromosomes around. So the next stage is metaphase. Here we're in metaphase 2. Chromosomes are going to line up along the equator of the cell, similar to what you would expect in metaphase. In anaphase 2, this is when you see your sister chromatids being pulled apart. So this is when we get the final separation of those duplicated chromosomes into individual chromosome strands. And then in telophase, each one of these 2 cells that went into meiosis 2 divides again, ending up with 4 non-identical haploid cell. So if you look at these, because of crossing over, you can see that this long blue chromosome does not look exactly the same in each of the cells that's produced on this left-hand side, because crossing over creates a little more genetic diversity. So all 4 of the haploid cells may be slightly different from one another. Mitosis and meiosis differ in 3 main ways. Mitosis is going to create diploid cells for the body of the organism, whereas meiosis creates haploid cells to be used in sexual reproduction. Anaphase in mitosis separates sister chromatids, whereas in meiosis, anaphase 1 separates homologous chromosomes and anaphase 2 separates the chromatids. And then mitosis creates 2 daughter cells that are clones or identical to the parent cell, whereas meiosis creates 4 unique cells from that one parent cell. To put this in the human standpoint, since we're all humans watching this video, in humans, spermatogenesis is the process that happens in human testes to create 4 sperm cells for every parent cell. Spermatogenesis begins at puberty in males and continues until death, but aging does affect the quantity and the quality of the sperm. The sperm are basically a nucleus with a tail. It's a DNA package with some mitochondria to power that tail, and we've got an illustration of some sperm here. In comparison, oogenesis, which is producing human eggs, this begins in human ovaries actually before birth. And the developing eggs are held at prophase 1 until puberty. And so when a woman or a female is born, all of the eggs are present that she is going to have, and they basically are in a paused nature until puberty occurs. At puberty, individual egg cells mature during each menstrual cycle, and they go basically to the end of meiosis 1 before ovulation occurs, before that egg is released. The egg that is released at ovulation is paused again. It's holding at metaphase 2, and meiosis will only be completed in an egg cell if fertilization occurs. Also, only 1 large egg cell is produced by meiosis in humans. It contains the haploid DNA and all of the cell organelles. The other 3 cells are basically just discarded bundles of DNA, and they have their own name for polar bodies. So you can see there's a big size difference here between an egg cell and the sperm that are just sort of laying on its surface here, waiting to get in for fertilization. So when you are inheriting your DNA, it's equally from parent, half from your mother and half from your father. But all the other stuff that makes up your first cell is coming from your mother. In fertilization for humans and for all organisms that do sexual reproduction, 1 gamete from each parent organism combines to create a new cell that is diploid in nature. It contains all of the DNA found in the original cell, and it has a special name as a zygote. The zygote then goes on to use mitosis to create all the new cells needed to build the organism. But some of these cells will actually specialize. They will change to become germ cells to be able to produce the gametes by meiosis needed by that organism in the future. And just have a little illustration on the bottom how a zygote starts as one cell and it divides and divides and it divides. And during this stage in development, there actually is not a growth period, so you can see how the cells are getting smaller as they divide, and then they form various layers. That's the start of specialization that goes on to create all the different organs and organ systems that are in a more complex organism. So that covers what I wanted to say about meiosis and the different stages that create these special haploid cells for sexual reproduction.